And now we will learn about the sessions, how the second part of the sessions will be day 2 and day 3. And before that, let me just quickly tell you that what it will be about. So again, thank you, Dr. Harpreet. Now let's learn about the topic and let's learn about what would be the pace of this. So that in this introductory workshop, our focus will be to give you an in-depth overview of important omics technologies that is changing the way healthcare is, and this entire system is rapidly evolving. So our focus will be to making you learn about these complex technologies with several different layers so that it becomes easy for you to learn, and you have ample amount of resources with you to take you which you can take on for the next 1 month to learn about all of this. So we will learn about obviously the next generation sequencing technologies and the data, the different omics layer data, and then we will also learn about how this it all integrates into drug discovery and how this is actually helping in the process of drug discovery. So we will start with genomics, like giving you an overview of genomics, that genomics is an essential subfield of bioinformatics, and it's a very important field now, which is a major force in expanding our knowledge about genetic association with with diseases and other traits. So it's an interdisciplinary field. So in such interdisciplinary field, we have an opportunity also to learn about other things. So the other things would be to learn about R and Python, how R and Python is used in genomics. What are the different technologies or different algorithms that we use in genomics to gain insights about different things, such as, multiple sequence alignment of genomes. Then we are talking about, DNA methylation factors. Then we are talking about massive databases such as GeneBank, EMBL, and SRA that allows us to do all of this research. So this will be focused on that, and this is what genomics is going to be all about in this workshop. And then the another technology that we are going to learn about. So there are three key technologies that we are going to cover in this workshop, along with chemoinformatics, is genomics, transcriptomics, and metagenomics. So transcriptomics is the process by which the information in a strand of DNA is copied into a new molecule of RNA. So this transcription is like a dynamic process, right? So the transcriptome is a snapshot of genes transcribed in a cell at a particular point of time. And this is very important to learn, that this is a complete snapshot of the entire genes at a particular point of time. So this actually enables us to gain a lot of information when we compare different stages, different conditions. So that's why we learn so much about transcriptomics, and that's why it is being used in so much in clinics and in research. So the sum total, that is what the transcriptome is all about. And then we are going to learn about different analytical steps. So how do you analyze, once you have the transcriptomic data, it's deposited in public repositories, and you can answer some good questions, some good biological research questions, like biomarkers and finding critical genes that are expressed at a certain point of time that gives you an idea about their function. So all of that can be done through transcriptomics. And what we are going to learn is that how do you do the processing. So from a raw data, so you will learn about the raw data on 26th December, and on 27th December we will be learning about how do you process this raw data into something meaningful. So processing, exploring, and we'll learn about gene and isoforms, downstream analysis, statistical analysis, and then interpretation of that kind of data. So that would be one of the key objectives of this workshop, to get you comfortable with the visual T-Bioinfo platform, to get you comfortable with the research that can be done using such tools and algorithms. Sorry. And the next thing would be, after transcriptomics, is also to learn about metagenomics. Not after, but we are going to learn about these simultaneously in a way that it all makes sense. So in metagenomics, we would be learning about microorganisms, and then the samples that are collected from the natural environment. And we will be learning about the community structure, because in a natural environment, you'll find all kind of species richness, and then you'll be investigating their functional roles and how these microbial communities are playing a key role in human health, human disease, and many sort of interactions that we are learning from this sort of data. So again, we will be going through some of the project examples, and I will take you in a few more slides the entire schedule of the program. So here we will be going through the gut microbiome project. We'll learn about that. We'll learn about brain access project, and then we will learn about how this is being useful for those who do not have any background of metagenomic technology, and people who do have a background can explore more further. There's like metagenomic sequencing, analysis, phyloseq, and all the biome analysis that one can do with the tools that we will be showcasing you, and with the resources, tutorials that you can take help from to do this on your own. So learning about the technologies, omics technologies, genetic data, and now you're looking for solution strategies. Now you're looking for molecules, you're looking into structure, you're into three-dimensional world. So from three-dimension to two-dimension, from two-dimension, that is the sequence, to single dimension to multi-dimensional data, and from multi-dimensional data we move to three-dimensional data. And again, from three-dimensional data, we will move to, again, matrices. So again, two-dimensional data to do data science and AI. So this is really a fascinating journey from different type of data types that we were going to learn about in this short workshop. It is just to give you that what all can be done within this space. So one of the things that is going to be covered is protein folding structure prediction, because when you think about these important biological processes, you think about also the diseases, you think about also what is available and what is not available. For a computational biologist and for a person who does not have all the resources to do the experiments, we kind of rely on predictions, and we do predictions all the time. We humans do it all the time. So we also have built systems to do better prediction. One of that system is Google's DeepMind's AlphaFold. So we will be learning about how to run AlphaFold and what are the applications of AlphaFold with different variants that you're learning about, how that influences the structure and the function. Now we are back to the schedule for actually the day 1 and day 2 after the orientation session on 26th. This is 27th and 28th, and we have sessions starting from 10:45 a.m. To 6 p.m. till 6 p.m. in the evening, and all of this will be recorded and will be shared for those who are missing out on any of the sessions. But it would be a great experience starting from the beginning till the end. We will start with the orientation of the program to the program. We will see if you have the learn profiles up, and we will see if you have the resources emailed to you and all of that you're prepared for the session to start and do some hands-on experience and learn from the experts. So we will start from introduction to NGS technologies, different type of technologies that I just described to you, by taking some examples and learning some of the pipelines, that what we can experience and what you can extract, and how do you run the pipeline, what are the different components there. So you're going to learn about genomics, metagenomics, and then transcriptomics with example pipelines. Then there's a lunch break. Obviously we all get tired, and after that after the lunch break, we will have the session on how do you go next after you run the pipeline. How do you do with that? What are the different outputs? What are the different analysis method, and how do you interpret that? That would be for 1 hour. And after that, we will have, so these sessions will be divided into, again, 2 sessions. So once for the first session, we will be learning about the mutation. We will be learning about genomic visualization. Then we will be learning about the mutation and variant analysis that you can do with genomics. So here you're learning about genomics. You're learning about application of genomics. Before that, you got an overview of all the omics technology, and you learn about different pipelines that you can use to analyze such data. After that, when we are done with around 2:30 p.m., so after that, we start with our specialization tracks, which is from 2:30 p.m. to 3:30 p.m. and this is Indian Standard Time, so you can change it according to your time zone. Now we have in that session we have application of NGS. So we will be learning about the TCGA project, and then we will be learning about some of the projects related to COVID-19 sequencing. So that is not mentioned here, and we have included that now. So wastewater management. So we will learn about 3 case studies in brief here, and then we will actually work on those case studies later in the last session. So here you will get an overview of what the case studies are all about and what are we going to do, and then we will have a tea break. So after the tea break, we will start with cheminformatics application and case study. We will go over some of the softwares that are being used in cheminformatics and give you an idea of how and what are the things that are that you can start with using some of the tools that are available online. Then we will start with the last session, which is a breakout session where you can choose that which session you want to you're more interested in. So is it like machine learning and drug efficacy and learning about projects that are available applying transcriptomics analysis or genomics analysis or structural analysis. So we will have 3 rooms, any of you which you can join, and we'll be having obviously the recording, so you can learn about the different sessions later as well if you're a registered participant. In the second day, we will go through the statistical analysis, because all of these technologies are producing you a table. So that table is again like a matrix. It has rows, it has columns, and it is a very big table. So how do you interpret that table? We will apply methodologies, algorithms such as DEGs, some understand about the beta diversity, and we learn about from the structural perspective as well. So after that session, when we are well versed with what all that you can expect from this kind of data, what is the way to learn how to extract meaningful data, because one of the challenges with big data analysis and being a data scientist is that you have to analyze big data, and then you have to find critical information from a huge amount of information. So that's really a challenge, because data annotation is again a very trivial thing. So we will learn about that in the next session, that how you can explore unexplored data, how which you don't have no idea about. That is unsupervised machine learning, and then supervised machine learning. And we have an expert with us on deep learning, Dr. Raghavendran, as you have heard from him. So he will be taking us from that different types of algorithms that are used for such analysis. And then from a biological standpoint, what all of this analysis is making sense. You need to learn about that, right? You need to learn that why we are doing this, why we are using machine learning, and from what we are getting from machine learning, and why it is helpful for biology. So that is the connection to the biology, downstream analysis, pathway analysis, how it is affecting the pathways, if there are mutations, how it is affecting how it is becoming cancerous, and how it is applicable for metagenomics. And then we have an industrial panel discussion, and we have presentations and discussions where we will be telling you about the opportunities that are upcoming. So internship opportunities or job opportunities, or the things that you need to be prepared for to get a job in the industry. We will be hearing from some of the industry panels coming from Prima's Life Sciences and Pine Biotech. And then we have the certification and Q&A and networking, where you can network with the community and discuss and do all of the interaction that can help you maybe get different opportunities that are there. So that's about the workshop, and that's about the entire curriculum about the workshop. And if you have any questions about the curriculum and all of that, I'm happy to answer to those questions. And now I will invite Ms. Sonalika, if she is around, to take you through the registration and this workshop registration fees and all of that, and also the resources that you will have. Thank you. Thank you, everyone.